Hello, I'm David Gonzalez, and I'm gonna talk about the Gregor Mendel life and what he did.
Gregor Johann Mendel, or Gregor Mendel, was born on 20th of July of 1822, and he died on 6th of January of 1884 on Czech Republic, and he was known as the founder of the modern science of genetics. At first, he experimented with pea plants. And with the pea plant, he started to discover how the genetics work in humans and in plants.
He at first started with reproducing the same plant because the plant is hermaphrodite, and he started to discover how it works, and then he started to do it with other types of plants.
When he was discovering this method, he at first started with plant head, pod shape and color, seed shape and color, and the flower position and color. With seed color, he showed that when a yellow pea and a green pea were bred together, their offspring plant was always yellow.
So, to explain it easy, he invented the recessive and the dominant heterozygous plant. The dominant heterozygous plant is the one that, when it reproduced with another plant, is the one that can have the more genetic information in the new baby plant. The recessive heterozygous plant is the one that, when it's reproduced with another plant, have the less genetic information to the next baby plant.
The punett square. The punett square is used to represent the inheritance of traits along generations. The first step to build a punett square is to draw a square and divide it into 4 equal spaces. Then, in this case, to build the punett square of the F1 generation, you need to put the parents. In this case, I'm going to use the P as the character that is going to represent the genes. The parents are double capital P, and the other parent is double lowercase p. The capital P's are going to be ubicated above the square, and the lowercase p's are going to be ubicated next to the square on the left side. Then you need to multiply the letters. The first case is going to be P capital by P lowercase. So the result is going to be capital P and lowercase p. To know how to describe the punett square is going to be very easy. You need to know that there are 2 types of description, the phenotype and the genotype. The genotype consists of 3 types: the homozygous dominant, the heterozygous, and the homozygous recessive organism. And on the phenotype, in this case, we are going to talk about the color of the flower trait are going to be the dominant is purple and the recessive is white. An homozygous dominant organism is going to be a double P organism on the punett square. An heterozygous is going to be a mixed organism that is going to be a capital P next to a lowercase p. Finally, the homozygous recessive organism is going to be a double lowercase p on the Punnett square. The phenotype is much easier because the purple or the dominant are going to be heterozygous and homozygous dominant organisms, and the white are going to be only the homozygous recessive organism. These are the basic steps to build a Punnett square. To build the another squares, you need to repeat the same process, but taking into account that you would have different parents and different results, taking into account the genotype and the phenotype. But if you want to build a two-trait Punnett square, you need to first make the same steps, that is building the square and locating the parents. And next I'm going to tell you what are the parents of the generation of the two traits. The Punnett square parents of the F0 generation of the Punnett square with two traits are going to be F0 are going to be, in this case I'm going to use the letters P and W. The F0 generation is going to be capital P next to lowercase p is as one parent. The other parent is going to be capital W next to a lowercase w. The F1 generation is going to be a multiplication of the two parents of the F0 generation. So as a result, you would have P capital and W capital. Another parent is going to be capital P next to a lowercase p. And another parent is going to be lowercase p next to a W capital. And finally, you would have a lowercase p next to a lowercase w. Okay, I'm going to explain you how to get F2 generation. The first and the basic way to identify the parents is going to be a double multiplication of the parents, because you have two letters and not one as the previous cases with the Punnett square of only one trait. The first spaces and the step you need to follow to complete the Punnett square is going to be PW and PW are the parents of the first two squares, taking into account that the one parent is horizontally ubicated and the another is vertically ubicated. So we are going to proceed. P by P is going to be double P capital, and the another parents are going to be multiplied. So as a final result on the first square, we need to get double capital P next to a double capital W. So if you complete this process, have all the Punnett square of the F2 generation with the traits completed. And then to identify the genotype and phenotype, you repeat the previous process held on this period.